Okay, we're discussing the Sarcomere model today. This here is the Sarcomere model. And around the outside, you will see the webbing called your Sarcoplasmic Reticulum. So here and here, Sarcoplasmic Reticulum. At the ends of your Sarcoplasmic Reticulum, you have your terminal cisternae. Okay, so on either side of this yellow band are your terminal cisternae. And the yellow band represents your T-tubules. So Sarcoplasmic Reticulum is this webbing, Sarcoplasmic Reticulum here. Then here and here, each of these four are going to be your terminal cisternae, and then the yellow here is going to be your T-tubule. So that's the outside covering of the sarcomere. Now let's pull this off, and you will see the inside of your sarcomere. So we are going to talk about the sarcomere itself. And remember, the sarcomere is the smallest functional unit of the muscle cell. And this is where the myofilaments are organized in an alternating pattern of thick, thin, thick, thin. And those alternating patterns of thick and thin are what give the muscle cells, or the skeletal muscle tissue, that striped, striated pattern. This sarcomere has a few parts to it. So first part is thick filaments, and the thick filaments are represented by these thick blue bands. It also has thin filaments represented by these red bands. It also contains stabilizing proteins, and it contains proteins that regulate the interactions between the thin and thick filaments. So it contains all four of those things. So again, remember, as we're going through this, the red are your thin filaments, and these thick blue ones are your thick filaments. Okay, the first thing we're going to look at are the Z lines. So your Z lines are going to be these red zigzag lines here. They mark one sarcomere. So from one Z line to the next Z line is one sarcomere. And then there will be this Z line to the next Z line over on my right side would be another sarcomere. And on my left side, from this Z line to the next Z line on my left-hand side would be a third sarcomere. And so they zip together and they form a long repeating pattern throughout the whole muscle cell. So again, these are my Z lines, and my Z lines are going to be where all of my thin filaments bind together. So all of these thin filaments are going to bind to the Z line. Now this line right here, this line is called the M line, I think in the middle, okay? So in the middle is your M line, and it's going to bind all of these thick filaments together. So the M line binds all the thick filaments, and the Z lines bind all of the thin filaments, okay? So those are the two lines that you have to know, the Z lines and the M line. Now, the next thing we're going to talk about are the bands. You have three bands that you need to know. You have the I band, the A band, and the H band, okay? And there's a little way of remembering it. Pull this up so you can see it. Okay, so the I band, you're going to think thin, because the I band has thin bands only, or thin filaments only. And so if you spell it little t, little h, capital I, little n, you see that the I stands out. So in the I band, there are thin filaments only, okay? The A band, you're going to think and, A-N-D, because this has thin and thick filaments, okay? So the A band has thick and thin filaments. The H band, if you're thinking H like heavy, the thick filaments will always be heavier than the thin filaments. So your H band has thick filaments only, okay? So it's just a little way of helping you remember what is in each band. Because you have to know this, remember, for your lecture and for your lab. So make sure you know what is in each band. So when we're looking at this model, this area right here and here, where there's only thin filaments, this is your I band. Because remember, I band, thin filaments only. So this is your I band. Now from here all the way into the middle, all of this is going to be your A band, because your A band has thick, the blue ones, and thin. So from one hand to one hand here, this is your A band. Now in the center here, you have thick filaments only, so this is going to be your H band. Remember, H, heavy, so you're thinking heavy, thick band, okay? So your thick filaments will be found here in your H band. Now this area right here where you have thin and thick, this is called your zone of overlap, okay? Because remember, your A band has the zone of overlap, plus it has your H band, okay? So just this area right here of the A band where both of them are overlapping each other is your zone of overlap. Okay, so again, I band, thin filaments only. A band from here to here, all of this is your A band, remember, thin and thick. And then right here is your H band, remember, heavy or thick filaments only. This area right here of the A band where you have the thin and the thick overlapping each other, this is going to be your zone of overlap. Okay. Now, with the Sarcomere model, we have the sliding filament theory. So when we contract a muscle, we have this, the thin filaments sliding over the thick filaments. So you can see that here. Now our muscle is contracted. When it relaxes, it slides back out. Contraction, it slides in. Relaxation, it slides back out. So this is called the sliding filament theory. Okay, and there are a few things that you need to know about this sliding filament theory. There are 5 things that occur. So we're going to pull it out here, and I'm going to show you each of these 5 parts of the sliding filament theory. The first part is when you have a contraction, the Z lines get closer together. So right now our muscle is relaxed, and we're going to now contract the muscle, and you'll notice our Z lines got closer together. Watch that one more time. This is relaxed, and we're looking at these Z lines right here. Now our muscle is contracting, and you'll see that our Z lines got closer together. So that's the first thing that occurs in the sliding filament theory. The next thing that occurs is that our H bands and our I bands get more narrow. So here are your I bands. What is that, about 1.5 inches, maybe 2 inches on either side when the muscle's relaxed. Okay, now the muscle contracts, and now my I band is just 1 finger width thick. Okay, so my I band went from about 1.5 inches to 2 inches to 1 finger width wide. So my I band got more narrow. Okay, now my H band. So my H band right now is like this. So it's like maybe 4 inches wide. But when my muscle contracts, now it's not even probably about 1 inch wide. Okay, so my H band became more narrow. Okay, so again, when I contract, my Z lines got closer together. And when it contracts, my I bands got smaller. Now they're only this wide. And my H band got more narrow. Okay, so those are 3 different elements of the sliding filament theory. Now let's talk about the zone of overlap. So our zone of overlap is probably about 2.5, 3 inches on either side. And as I contract, my zone of overlap is now like 4 or 5 inches wide on either side. So my zone of overlap widened, okay? So again, my I band became more narrow, my H band right here became more narrow, and my zone of overlap became wider, okay? Now the last thing is the A band. Remember, our A band is from here to here where you have the thin and the thick overlapping. Okay, but watch this. I'm just going to use this side so I can move it. I never moved this hand, okay, because I never moved these thick filaments. So that where the end of those thick filaments are, that's your A band. your thick filaments will never move. So your A band never changes. So your A band remains constant through the entire contraction relaxation cycle, okay? So again, the 5 parts of the sliding filament theory, your Z lines got closer together, your I bands became more narrow, your H band became more narrow, your zone of overlap became wider, but your A band remained constant. Okay. And again, this is your Sarcomere model. The red are your thin filaments, the blues are your thick filaments.